pathophysiology of a disease. We're trying to figure out how that disease causes symptoms. So in the case of herpes, or the herpes simplex virus, we're trying to figure out how the virus infects cells and causes symptoms. So let's take a look at this skin cell over here. This is a cell that may live on your lips. And here's the nucleus. I'm going to draw it dotted so we can see what's going on inside. And in there's going to be some DNA. And I'll draw it like this, even though DNA is a lot more tightly wound than it is here. Then I'll label these things. So this dotted line here, I mentioned, was the nucleus. Or we can also refer to it as the nuclear envelope. And then on the inside, this guy is our cell's DNA. It's our own DNA or our nucleic acid that lives in there. Now let's imagine this cell is going to be infected by the herpes virus, so I will use this triangle to symbolize herpes. And so this triangle, or this virus, will come and attach onto the cell membrane here. And within the cell, the herpes has its own DNA, the viral DNA that exists, as well as some proteins that are in here. And I'll explain in a few minutes what these proteins help it do. But just realize first off, this green border, the triangle on the outside, is a nucleocapsid. This helps the virus inject the DNA and the proteins into the cell, like we'll see in a minute. Right here we've got some proteins, as I'll mention in a few minutes. These help incorporate the viral DNA into the human or host DNA. And finally, this yellow strand right here is the viral DNA. So this is the DNA of the herpes simplex virus. And so our first step here is that the nucleocapsid has proteins that help inject the viral DNA and these proteins into the human cell, which means that, as I'll draw right here, is to inject viral DNA. So once we do that, let me just draw our cell again over here. Now the viral DNA will be inside of the cell, and with it will be these viral proteins that the nucleocapsid injected as well. So at this point, the viral DNA will use these proteins to inject itself or incorporate itself, which is a better word, into the human DNA here. And so our next step is that we will incorporate viral DNA using these proteins. And so what you'll end up getting, and I'll bring in another sample of the cell right here, so this is the cell we've been working with, is the human DNA here, but now it's also attached to the viral DNA. Now at this point, this cell that now is under the control of the herpes virus, because the viral DNA is now in the brain or the nucleus of the cell, this cell can do two things. And I'll draw that right here. Here's one path that we can take, and then here's another path we can take. So I'm trying to draw all of this like a cycle, so let's complete this cycle and talk about this step right here, which after we've incorporated the viral DNA, we will start to mass produce the herpes virus, which, as I'll draw this cell again over here, you'll begin to see a lot of these nucleocapsids being formed again, and so they're going to swell up in the cell. These guys will also have their own copies of the viral DNA, and you might not be able to see it, but I'll just mention the protein is there as well. And in fact, this occurs to such a crazy extent that the cell can't handle that type of viral production. And so what'll end up happening is that the cell will rupture. It'll pop open. So I drew this cell here, but let's draw another one up over here. And I'll really exaggerate this. There's going to be so many of these viral nucleocapsids that are filled with DNA and proteins that the cell will not have any more nutrients for it to fortify its cell membrane or for it to grow on its own. And so what will end up happening is that the cell membrane will fracture open like that. It'll pop, it'll burst, and you'll begin to have an exodus of these viruses out here, which then allows you to restart the cycle. And because the cell popped open here or ruptured, another term for rupture is lysis. The name of this phase for the herpes life cycle is the lytic phase. This is the lytic phase. And that's in contrast with what's happening here. Instead of killing the cell, what you'll see happening is that the herpes virus will remain dormant. And so what you'll get are two of these cells. So there's one here, and here's another one. And so the virus just kind of takes a back seat and allows the human cell to grow on its own. And so here, the virus will reproduce with the cell. And this relatively more dormant phase of the virus or the herpes virus life cycle is referred to as the lysogenic phase. And that's just to emphasize that the cell is able to undergo the lytic phase or undergo lysis of the cell. That's why lysogenic, meaning able to cause. So it has that potential, but is currently not doing that. It's just going along for the ride. And for the time being, the herpes virus is dormant, meaning it's at rest and not mass-producing its viral nucleocapsids, proteins, or its DNA. Now, an important thing about herpes is that it likes to be dormant in nerve cell bodies. And the term for nerve cell bodies is ganglion. So I'll write ganglion here. Or if you're talking about multiple ganglions, there's no such word as ganglions, so instead we say ganglia. And in fact, we know what specific ganglion or ganglia herpes likes to go into the lysogenic phase in, and that depends on where it occurs. So remember, herpes labialis is herpes that occurs on the lips, on the inside of your cheeks, or even on the tip of the tongue, so it's oral herpes. That tends to go and become dormant in the trigeminal ganglion. The trigeminal ganglion, which is a cranial nerve that helps you feel touch on your face. So that's for herpes labialis. Now, for herpes genitalis, there are several ganglia that the herpes can become dormant in, and we group them together and call them the lumbosacral ganglia, which is a reference to the spinal cord. It can be in the lumbar or the sacral region of the spinal cord, so somewhere in there. But recall, because this is lysogenic, we're able to cause lysis. These cells are able to pick right on up and enter the lytic phase. And there are a couple of things that are known to cause that. Sunlight is one thing that's been noted in some people to induce the lysogenic cycle to convert into the lytic cycle. Decreased immune function is another known cause. But what these all boil down to is that anything that's an overload of stress, and that can also include emotional stress, can trigger a lysogenic virus to enter the lytic phase, which is such an interesting concept because the herpes virus must be able to read how the cell is surviving relative to its environment. So it has become one with the cell. And unfortunately, once that cell is overly stressed, it's time for the virus to escape and reproduce and move on to another cell that'll be able to house it and help it grow.